let's finish up the chapter, section 5, nucleic acids. Nucleic acids are the polymers of nucleotides. Now there are two different types of nucleotides. DNA, which is deoxyribonucleic acid, which is found in the nucleus of our cells, the genetic material that stores information for its own replication and for the sequence of amino acids in proteins. So DNA is the directions to make protein that stays inside the nucleus. Our other type of nucleic acid is RNA, or ribonucleic acid. It performs a wide range of functions within the cell, which includes protein synthesis and regulation of gene expression. So DNA stays inside the nucleus. RNA is like a half copy of DNA that can travel from the nucleus into the cytoplasm, into the cell to do what needs to be done. So DNA is like the boss man, and RNA is like the messenger. Right? And they're both made up of nucleic acids. Each nucleotide has 3 parts. The first is a phosphate, just a regular phosphate molecule, a pentose sugar, depending on what nucleotide we're talking about, it's a different sugar, and a nitrogen base. And there are 5 different types of nitrogen bases. 4 of them are found in DNA. They are adenine, guanine, cytosine, and thymine, whereas RNA is made up of adenine, guanine, cytosine, and uracil. So uracil and thymine are the ones that are only found in either DNA or RNA. Now these nucleotides, which is made up of all 3 of those parts, are joined together by a series of dehydration synthesis reactions to form a linear molecule called a strand, which is a sequence of nucleotides. So each nucleotide has a phosphate, a sugar, and a nitrogen base, and they're bonded together to make that nucleotide, and then those nucleotides are bonded together in a long strand. So here's what an individual nucleotide looks like. Here's our phosphate, our pentose sugar, and our base containing a nitrogen atom. So here is deoxyribose versus ribose. Deoxyribose just has 1 less oxygen. That's all it is. Ribose has an oxygen right here, whereas deoxyribose does not. That's the only difference in the sugar molecules. Here are the different nitrogen bases that are found in nucleotides. Here's cytosine, thymine, uracil. These are in a category called pyrimidines. And these 2 over here, adenine and guanine, are purines. Pyrimidines are made up of 1 single ring that consists of a mostly carbon backbone. Purines are 2 rings that are bonded together. And whenever we get to DNA replication, we'll talk about why that's important. For right now, I just want you to know that there are pyrimidines and purines and which category they are in. I don't need you to know the why yet. I just need you to know it. And here's a diagram showing all of those diagrams on one slide. The structure of DNA and RNA. The backbone of the nucleic acid strand is composed of alternating sugar-phosphate molecules. RNA is predominantly a single-stranded molecule, whereas DNA is double-stranded, and you'll see a picture of that on a later slide. And DNA is composed of 2 strands, held together by hydrogen bonds between the nitrogen bases, and they twist around each other, forming a double helix. The nucleotides may be in any order on the strand, but if there's an adenine on one side of our DNA, there's always going to be a thymine on the other side of the DNA. If there's a cytosine on one side, there's going to be a guanine on the other side. This is what's referred to as complementary base pairing. So we know that the number of adenine and guanines will always equal the number of pyrimidines, the thymines and the cytosines, because they complementary base pair. So here is a table to help you remember the differences between DNA and RNA. It talks about the sugars that are there. Remember, deoxyribose is found in DNA, ribose is found in RNA. Thymine is found in DNA, whereas uracil is found in RNA. DNA is double-stranded, RNA is single-stranded. And DNA is usually found in that helix shape, that twisted ladder shape. RNA is not in that shape. So here's the structure of RNA. Here's our sugar, phosphate, sugar, phosphate, sugar, phosphate backbone. And here we have our nitrogen bases. Here's our DNA structure. Here is the sugar phosphate backbone, here in blue. Notice it's twisting. That's our helix shape. It's called a double helix because there are two strands. You'll notice that our nitrogen bases are here in the middle. And these little dots right here represent our hydrogen bonds holding those nitrogen bases together. Adenosine triphosphate, also called ATP, is a nucleotide composed of adenine and ribose and 3 phosphates. ATP is a high-energy molecule due to the presence of that last phosphate. Those 2 phosphates are not happy being there, so they can easily be broken off. Hydrolysis, the breaking off of that terminal phosphate, that last one, causes it to now be known as adenosine diphosphate, meaning 2 phosphates, and just a phosphate. When that energy is released, it can be used to do work in the cell. The hydrolysis of the ATP molecule can be coupled with chemically unfavorable reactions in the cell to allow the reactions to proceed. So if the cell needs energy for something, it's going to go get an ATP molecule, it's going to break off a phosphate and use that energy the way that it needs to. Key steps in the synthesis of carbohydrates and proteins use ATP, as does muscle contraction and nerve impulse conduction. Because ATP is used in all of these ways for energy production, it is called the energy currency of the cell. So here's what an ATP molecule looks like. Notice this ADP plus energy. This energy is the energy that's getting used. And that's it, guys. If you have any questions, let me know.